This is for our genetics lab, or Snoogie's lab. So I have two plastic Easter eggs here. One that says M, so this is your male egg. And this one says F, that's the female egg. So we're going to open them up and see which alleles we have. You guys just don't get to eat the candy, unfortunately. I had to tape some of these shut so that the candy wouldn't fall out. So give me a second here. Gotta get off the tape. Okay. Here are male alleles. These might not necessarily be in the same order as they are on your paper. I'll put the two candies together. Ooh, got bubbles rolling away. Alrighty, so you have a green and an orange lifesaver. These are the Skittles. Yeah, they're two purple Skittles. A red and a yellow Mike and Ike's. You know what? Actually, hang on a second. My bad. I mixed up a Skittle. Let's try that again. So you have an orange and a purple Skittle, and an orange and a purple M&M. My bad. I should have paid attention. A red and a blue gum. The gumdrops are red and green, and the Starburst are yellow and red. So you have a bunch of heterozygotes here. Pull it out like this. So you are going to look at your paper or your lab notebook and write down, this is the male, so in one of those columns, like where it says Snoogie parent number 1, you're going to write out all of those traits. You can, if you want to, write the colors and then come up with letters if that helps you. For the sex, it would be male, so it's XY. Now let's open up the female egg. Whoa, I throw it across the room. Okay. I'll try to put them in the same order for you guys here. Ooh, these look like all homozygous ones. How the heck did that happen? I promise I didn't do it on purpose. Well, except for one. Okay, this time, we have two red lifesavers, a purple and an orange Skittle, two yellow Mike and Ike's, two blue M&Ms, two blue gumballs, two green gumdrops, and two pink Starbursts. I'll zoom that out so you can see them. And for its sex, this one was the female egg, so you just write XX. There's your female egg. Now what you need to do is make letters for each of these and then do genetic crosses. We realized in class that it's easier to do them all as individual Punnett squares, so for right now, you can just forget about the linkage part that I wrote in there because that kind of makes the math hard. You decide, do you want to do a Punnett square or do you want to do the math or a little bit of both? You decide which one makes more sense to you. So for example, if I were going to cross my lifesavers, here are my female lifesavers. Here are, whoa, one was running away. My male lifesavers. So you could literally do a Punnett square kind of like this, where you just come up with your own letters. Or if you want to do math and figure out dominant recessive that way, that would work. So in that, I want to say it's the fourth table, where you're writing out the possibilities of each different offspring combination. And that one, you would write down like half, 0, or a quarter, half, a quarter, or 100, 0, 0, whatever the chances are of having that. I will find a separate video up with the paper on it to kind of give you some examples of that. If you have any questions, please let me know.